Okay, now we start looking at our local and action potentials and compare them. The local potentials, remember, travel short distances, and the action potentials are going to travel the entire length of the axon. So your local potentials are graded. They produce variable changes in the membrane potentials, but your action potentials will cause maximum depolarization to 30 millivolts. Now the all or none principle applies here. It either happens completely or it doesn't happen at all. So if the neuron doesn't polarize to threshold, then you're not going to have an action potential. But if it gets to threshold, it's going to go all the length of the axon. The action potentials are not dependent on the strength of the signal or the frequency or the length of the stimulus like local potentials are. So it's not graded. You got to figure out how to move to the next slide. The local potentials are reversible. When that stimulus ends, the neuron goes back to resting potential. But the action potentials are irreversible. Once it reaches threshold, it can't be stopped. It's going to go all the way to the end of the axon, all or none. The signal distance is greater for action potentials because a local potential only happens in a small area of the axon. So the local potentials are decremental. They decrease in strength over a very short distance. Action potentials are non-decremental. The signal strength doesn't decrease even though it goes all the way to the end of the axon. The action potentials have to be conducted or propagated to get along the entire length of the axon. They are self-propagating. They only go in one direction. Each action potential will trigger the next section of the axon. So it starts at a trigger zone and it goes all the way to the axon terminal like dominoes. It only travels in one direction. And so the sodium ion channels will open and it continues down like dominoes so that the next section depolarizes. And the action potential propagation all the way down the length of the axon is what we call a nerve impulse. Now the speed of the conduction is the rate of propagation. This will be influenced by the diameter of the axon and whether or not it's myelinated. Axons that have a larger diameter have faster conduction speeds because the larger diameter decreases resistance to conduction. Also, whether or not there's myelin. That will influence what type of conduction. There's two types. There's saltatory and there's continuous. Saltatory conduction is only in myelinated axons because you've got that insulating property of the myelin, so that it will only depolarize at the nodes of Ranvier. So it's going to depolarize here, and then it'll depolarize here. So it's going to jump from one node of Ranvier to the next. It doesn't have to go through the entire axon. It just jumps over the inner nodes. That's saltatory conduction. Continuous conduction is what happens in unmyelinated axons, where every single section of the axonema from the trigger zone all the way to the axon terminal has to be conducted, has to propagate. So that slows it down. It happens here, and then it's going to happen in the next section, and then it's going to happen in the next section. So it's going to move section by section all the way down that axon. And so the whole axon, the whole axonema has to depolarize. So it's a lot slower. Now our neurons have to communicate with other cells. They communicate with other neurons and other cells. The synapse is where the neuron meets its target cell. And so we call it a neuronal synapse if it's going to communicate with another neuron. And this synapse can either be electrical or it can be chemical. So it occurs between an axon of one neuron and another part of another neuron. The transfer of the chemical or the electrical signal between neurons at that synapse is called synaptic transmission. And this is what allows voluntary movement, cognition, sensation, and emotions. An average presynaptic neuron, the one before the synapse, will form synapses with as many as 1,000 postsynaptic neurons, the ones that are after the synapse. And the postsynaptic neuron, the one after the synapse, can have as many as 10,000 connections with presynaptic neurons. So it can have 10,000 presynaptic neurons communicating with it. The electrical synapse will occur between cells that are electrically coupled by gap junctions. The axolemas of each neuron in the synapse are nearly touching. And these gap junctions that are like the straws will align the channels and form pores so that ions can flow through. We find this in the brain and any area that's responsible for programmed automatic behavior, like breathing. We also find them outside the brain, in your cardiac muscle tissue and your smooth muscle, so that you have some coordinated muscle activity. The electrical current flows directly from the axoplasm of one neuron to the next. And it creates two unique features of the electrical synapse. First of all, the transmission is bidirectional. Either neuron, the presynaptic or the postsynaptic, can be presynaptic or postsynaptic, depending on the direction that the current is flowing. And it's nearly instantaneous transmission. It will depolarize a lot faster, like 0.1 milliseconds, as compared to chemical synapses that take at least 1 or more milliseconds. So it's a lot faster. And when you look at it, you can see what I'm talking about. See how they have these gap junctions here so that the ions can flow through easily? Just flow through real easily, either direction that they want to flow through. Now your chemical synapses, this is the majority of the synapses in the nervous system. And they're more efficient than the electrical synapses are. They don't lose any signal strength at all. To compare the two, the electrical and chemical synapses, there's three differences structurally. First of all, the synaptic vesicles are filled with chemical messengers or neurotransmitters in a chemical synapse, and that will transmit signals from the presynaptic to the postsynaptic neuron. They also have a synaptic cleft, a small extracellular fluid-filled space that separates the presynaptic from the postsynaptic neuron. This is in the chemical synapse. Remember, we have gap junctions to connect the two neurons in the electrical synapse. Also, the postsynaptic neuron has these neurotransmitter receptors that bind to the neurotransmitter that is secreted from the presynaptic neuron and that has diffused across that synaptic cleft. You're going to have a time gap or a synaptic delay from the time that the action potential arrives at the axon terminal and it has an effect on the postsynaptic membrane. Our chemical synapses are unidirectional. They are not like the electrical synapses that are bidirectional. So they allow for variable signal intensities. And more neurotransmitter is released from the presynaptic neuron, and that will cause a stronger response in postsynaptic. Whereas that's not true in in the electrical synapses, because the signals are always the same size. So you can see the chemical synapse here, how there's the gap between, in that synaptic cleft there, you've got this gap between the two. And you also see how we have these receptors here where the neurotransmitters are going to bind in the chemical synapse, and we didn't have that in the electrical synapse. Now, at the chemical synapse, it's a lot more complicated because there are multiple neurons that are secreting different types of neurotransmitters. They may be excitatory or they may be inhibitory. First of all, the action potential in the presynaptic neuron triggers the voltage-gated calcium ion channels in the axon terminal to open. And then all the calcium ions coming in will cause those synaptic vesicles to release that neurotransmitter into the synaptic cleft by exocytosis. And thirdly, the neurotransmitters then bind to the receptors that are on the postsynaptic neuron. And that causes the ion channels to open and leads to a local potential that, if threshold is reached, it'll go all the way up to a action potential.